If you've ever tried to map the complexity of disease,
Hmm.
cancer, infections, really anything, you realize pretty quickly that the goldmine is historical clinical data. We're talking huge biobanks, just decades of patient history stored away.
Absolutely. And almost all of that history, it's preserved as these formalin-fixed, paraffin-embedded blocks, FFPE blocks. They're absolutely invaluable. They're the physical records, right?
Yeah.
Diagnosis, patient outcomes, it's all there.
But they're also, let's be honest, a nightmare for modern genetic research. That formalin, while it's great for preserving the tissue structure, it's just brutal on the molecules inside.
Uh-huh. It shreds the RNA, causes breaks in the strands, cross-links proteins and nucleic acids, basically locks everything down.
Making it incredibly hard to get that critical genetic information out.
Exactly. For years, high-resolution methods, especially spatial transcriptomics, they've really struggled with FFPE. The RNA is just so degraded, so fragmented, that the signal you get is often weak, biased, or, frankly, unusable.
So you've got this huge bottleneck. All this vital clinical context, essentially inaccessible to our best sequencing tools.
Well, until now. That bottleneck is starting to break open.
And that's what we're diving into today, the technology that's unlocking these archives, Stereo-seq V2.
Yeah, our mission today is really to walk you through the core innovation in V2, how it manages to do high-resolution spatial mapping of total RNA, even on these really old, difficult FFPE samples.
And why capturing total RNA is the key.
Right. It reveals things we just couldn't see before. Non-coding RNAs, host-pathogen interactions, even detailed spatial immune responses. It's a really important step towards, truly personalized medicine based on these archives.
Okay, let's unpack that core technical innovation then, because this is really where the breakthrough happens.
Yeah.
If you've been following spatial biology, you know the previous techniques, even the first version of Stereo-seq, they were mostly built for fresh frozen samples, right? Pristine stuff.
That's right. And critically, those older systems almost all relied on capturing the polydenylation tail, poly-A tail, of messenger RNA.
Okay, let's break that down. So RNA is like a message. Why does looking for the end of the message, that poly-A tail, fail when the RNA is old and damaged, like an FFPZ?
Think of an mRNA molecule like a sentence. The very end of that sentence, the 3' end, most mRNAs have this long string of A bases, that's the poly-A tail. Traditional methods use a probe made of T bases, a poly-T primer, that sticks specifically to that tail. It's like finding the period at the end of the sentence.
Okay.
But in FFPE samples, that fixation and that aging process, it just chews up the RNA. Fragmentation, degradation, and that fragile poly-A tail. It's often one of the first things to get chopped off.
So if the tail's gone, your poly-T probe has nothing to grab onto.
Precisely. And that whole RNA message is lost to the analysis. You just don't capture it.
Right, so V2 basically says, okay, forget looking only for the tail. It changes the whole search strategy.
It does. Stereo-seq V2 makes this really key upgrade. It switches from poly-T primers to random primers. Specifically, they use random hexamers, 6N primers.
Meaning primers just 6 bases long, completely random sequences?
Exactly. Instead of targeting just that 3' tail, these short random primers can land pretty much anywhere along the length of any RNA fragment that's present. They hybridize almost discriminately.
So they capture everything, total RNA, not just the mRNAs with intact tails?
Correct. It allows for total RNA capture, which neatly sidesteps the whole problem of the missing tails and the severe fragmentation you see in FFPE.
And what's really interesting is they manage this chemical switch while keeping the incredible resolution from V1.
Absolutely. That was crucial. V2 inherits that ultra-high resolution. The capture spots, these DNA nanoballs or DNBs, are still spaced just 500 nanometers apart.
Which is tiny, like subcellular level resolution, potentially.
Theoretically, yes. You're looking at resolution fine enough to potentially distinguish signals within different parts of a single cell, not just assigning a signal to the whole cell or a block of cells. And of course, the V2 protocol wraps this capture chemistry and all the necessary prep steps for FFPE deparaffinization, rehydration, crucially, the chemical decross-linking to undo some of that formalin damage. It makes it a robust, end-to-end workflow for these clinical samples.
Okay, that makes technical sense. But switching to random primers, did that introduce any issues? Like, are you capturing a lot more noise, maybe? Ribosomal RNA, things you don't necessarily want, compared to the targeted poly-A method.
That's a fair question. And yes, by definition, random priming captures all RNA, including abundant ribosomal RNA, which often isn't the primary interest. But you have to weigh that against the massive bias introduced by degradation in FFPE samples when using poly-A methods.
Meaning the poly-A methods are biased towards the few RNAs that happen to have their tails intact.
Exactly. And they're biased towards the very 3' end of those transcripts. V2, by using random primers, achieves something much more valuable for FFPE. Remarkably uniform coverage across the entire gene body, from the 5' end to the 3' end.
Because the primers can land anywhere on the fragments.
Right. The gene body itself is much, much longer than the poly-A tail. So even if the RNA is fragmented, there are vastly more places for a random primer to bind along the body compared to just that one spot at the degraded 3' end. You get a much more complete representation of the original transcript, even if it's in pieces.
So you solve the degradation bias and get a fuller picture.
Okay.
What about the type of information? When you switch to capturing total RNA, what new things suddenly pop up on the map?
This is where it gets really exciting. You gain significantly enhanced sensitivity, especially for RNAs that don't have a poly-A tail to begin with, the non-coding RNAs, or ncRNAs. These were largely invisible to previous methods.
Right, the regulatory molecules, the functional RNAs that aren't messenger RNAs.
Precisely. So in tests on FFPE mouse brain tissue, V2 detected a massive number of genes, 42,440. Now, a lot of those were protein coding, sure, about 20,720. But what was striking was that nearly 23,500 of the detected genes were unique to V2 compared to poly-A methods run on similar tissue.
Wow, over half were new finds.
Yeah, a huge chunk. And this included many non-coding RNAs like SNRNAs, things like GM22980, SNORD38A. These are molecules involved in RNA modification and regulation, things we're only beginning to understand fully. V2 is essentially opening up that whole non-coding dimension in FFPE tissues.
So accessing the sort of dark matter of the transcriptome in these archived samples.
That's a good way to put it.
Yeah.
And this sensitivity ties into the next critical point, precision. If you're capturing all these tiny fragments, how do you make sure the signal you detect at, say, spot 1 corresponds to RNA that actually originated right there and hasn't just drifted over from spot 2? That's the issue of molecule diffusion.
Right. If the RNA fragments float away before being captured, your super high-resolution map gets blurred, like trying to read smudged ink.
Exactly. And the data showed V2 had significantly improved diffusion performance compared to even V1. They measured this using a metric called LWHM, left width at half maximal. Basically, it measures how sharp the signal boundary is between different tissue regions.
Okay.
Think of the edge between, say, the cortex and the hippocampus in the brain. With older methods, the signal for a gene highly expressed in the cortex might sort of bleed or blur across that boundary into the hippocampus area. V2 showed a significantly smaller LWHM distance. The signal drop-off at the boundary was much sharper.
Meaning the signals are more tightly locked to their original location, less blur.
Much less blur, more precise spatial localization.
And that precision, combined with capturing all that extra information, that's how they could identify 34 distinct cell types just within the FFPE mouse brain. That sounds like a huge jump.
It is. It moves way beyond just identifying broad anatomical regions, which is what you might get with lower resolution techniques, perhaps like Visium FFPE, which uses larger capture areas. V2 allows you to resolve distinct cell populations based on their unique expression profiles, even within complex degraded tissue. It enables genuine single-cell level insights from these archives.
Okay, this is where it gets really impactful. Let's talk clinical application. Moving from the mouse brain to real-world human samples specifically, triple-negative breast cancer, TNBC. These are FFPE blocks potentially stored for years.
Right. This was the stress test. They took 10 TNBC FFPE samples, and some of these have been archived for up to 9 years. And when they measured the RNA quality, it was rough.
How rough? You mentioned metrics like RIN and DV200. Can you give us a sense of what those numbers mean here?
Sure. So RNA integrity number, RIN, gives a general idea of degradation. But DV200 is maybe more telling for FFPE. It measures the percentage of RNA fragments longer than 200 nucleotides. In a good fresh sample, you want a high DV200, maybe 80 or 90%.
Meaning most fragments are reasonably long.
Yes. In these 9-year-old clinical samples, the DV200 values were sometimes down to 18.
18. So only 18% of the RNA fragments were longer than 200 bases. That's incredibly degraded.
It's extremely degraded. Over 80% of the RNA is chopped into tiny pieces. That level of degradation would typically kill any standard sequencing attempt, especially RNA-seq that relies on longer reads or intact tails.
But V2 handled it
remarkably well. They found that V2 maintained consistent gene capture efficiency across these samples, regardless of how low the DV200 score was. It suggests the performance is really quite tolerant to this severe RNA degradation.
That's resilience. So it can actually read the molecular story even in these really challenging samples. What kind of spatial insights did they pull out from these TNBC cases?
Well, right away, they could correlate the molecular data with the actual tissue structure, the histological heterogeneity. For instance, in one specific sample, T22301x78, the standard H&E stain clearly showed a necrotic area, basically, a region of dead or dying tumor cells. V2 spatially mapped the expression and found high levels of the gene, SPP1, concentrated precisely in that necrotic zone. SPP1 is known to be expressed by tumor-associated macrophages, suggesting V2 was pinpointing a specific immune niche, likely macrophages responding to the dying cells in hypoxic conditions right there in that dead zone.
Wow. Linking a molecular pathway inflammation, macrophage activity directly to the physical state of the tissue 9 years after it was preserved. What about the underlying cancer genetics? Could they see that too?
Yes. They use computational methods, specifically an algorithm called infer-CNV, which tries to infer copy number alterations, or CNAs, from the RNA expression data.
So looking for regions of the genome that might be amplified or deleted in the cancer cells.
Exactly. And they found distinct CNA patterns that allowed them to spatially segregate the tumor tissue into different subtypes, different clonal populations coexisting in the same tumor.
That heterogeneity is key in cancer.
It really is. And importantly, they pinpointed one tumor subtype, spatially distinct, that harbored a significant amplification right around the ESR1 gene locus on chromosome 6.
ESR1, that's the gene for the estrogen receptor. But this was triple-negative breast cancer defined by lacking that receptor, right?
Correct. That's what makes it intriguing. Finding an amplification of the gene locus itself, even if the protein isn't being expressed or detected by standard methods, spatially restricted within a TNBC sample. It points to potentially complex genetic drivers in that specific subregion of the tumor. It's a level of detail completely lost in bulk sequencing where you just grind up the whole tumor.
Okay, and because V2 captures the whole gene body, not just the ends, they could look at other things too.
Right. They leveraged that whole gene body coverage to analyze alternative splicing, or AS.
Which is how one gene can produce multiple different protein versions by sort of mixing and matching its exons, its coding segments.
Exactly. Subtle changes in splicing can dramatically alter protein function. V2 detected almost 1,500 different alternative splicing events within just one of these TNBC samples. And they highlighted one specific event, a skip exon, or SE event in the GIPC1 gene.
GIPC1 is a known player in breast cancer, isn't it?
It is, and this particular spliced form, the one missing a specific exon, was found to be significantly upregulated only in the tumor regions, not the surrounding normal tissue. So it acts like a spatially defined molecular marker, potentially linked to the cancer phenotype right there.
Incredible detail from such old, degraded tissue. Okay, let's shift gears slightly. From cancer to infectious disease, this total RNA capture, it sounds like it could be powerful for studying infections too, where you have both host and pathogen RNA mixed together.
Absolutely. That's where the host pathogen transcriptome profiling really comes into its own. Because V2 isn't biased towards just human polyRNA, it can simultaneously capture and map RNA from both the host, in this case, a mouse model, and the pathogen, Mycobacterium tuberculosis, MTB.
The bacteria that causes TB.
Right. They looked at lung sections from mice infected with MTB, sampled at different time points, 1 day, 4 weeks, and 8 weeks post-infection, PI.
And could they track the pathogen spatially?
Yes. They could map the MTB RNA reads directly onto the lung tissue structure. They saw that the MTB RNA signal peaked at 4 weeks PI, which matched the expected peak of bacterial load, and then it decreased by 8 weeks PI as the immune system presumably started to control the infection.
So what was the host immune system doing spatially, according to V2?
That was the next step. At 8 weeks PI, when the infection was being controlled, they looked specifically at the host genes, whose expression was spatially correlated with the remaining MTB hotspots, where the bacteria were still present, which host genes were firing up nearby.
Good.
The analysis showed a strong enrichment for genes involved in the adaptive immune response. It provides this beautiful spatial confirmation. The adaptive immune system, the B cells and T cells, are being actively recruited and engaged right at the sites of persistent infection.
Which leads us to what might be one of the most groundbreaking aspects of this, spatial immune repertoire analysis, studying the B cells specifically.
Exactly. To understand how B cells are responding to a pathogen or a tumor, you need to sequence their B cell receptors, the BCRs. These are the antibody molecules they use. The genetic sequences that define the BCR specificity are located near the 5' end of the RNA transcript.
Uh-oh. The 5' end. That's the opposite end from the poly-A tail, the end that traditional methods really struggle with, especially in fragmented RNA.
Precisely the problem. Poly-A capture gives poor coverage of the 5' end, but V2, with its random priming covering the whole gene body, gets much better, more uniform coverage, including that crucial 5' region.
Allowing assembly of the BCR
Yeah,
sequences.
enabling high-efficiency assembly of the variable regions that define the BCR clone. And in that MTB mouse model at 8 weeks PI, they found something really striking. The diversity of BCR clones and the frequency of mutations within those clones, a sign of adaptation, were both significantly higher in areas closer to the MTB infection sites.
So the B cells are literally evolving and refining their attack strategy right there on the front lines, next to the enemy.
It's direct spatial evidence of affinity maturation, the process where B cells fine-tune their receptors to bind the target better happening in situ, right within the infected lung tissue microenvironment.
That's amazing. And did this hold up in human samples?
It did. They validated this in human lung samples from TB patients. Again, using V2, they mapped the BCR clones around the necrotic granulomas the structures formed during TB infection, and they saw the exact same spatial gradient. As you move closer to the boundary of the necrotic core, the diversity of BCRs went up, and the mutation frequency within those BCRs went up.
Incredible consistency. The therapeutic potential seems huge here. If you can pinpoint the specific BCRs, the specific antibodies that are evolving right at the site of disease.
That's the hope, and they took it one step further. By comparing BCR sequences across 3 different human TB patients, they identified 16 shared recurrent BCR clones, clones with very similar sequences appearing independently in different people. And here's the kicker. These shared clones were found at a significantly higher frequency in the active TB patients compared to healthy control donors.
Meaning these are likely the B cells, the antibodies, specifically targeting MTB, the immune system's most wanted fighters against TB.
It strongly suggests that yes, these could be highly specific, potentially neutralizing MTB targeting BCRs. Finding these provides crucial leads, real molecular targets, for developing new diagnostics or perhaps even antibody-based therapies for TB.
It really feels like a paradigm shift. Stereo-seq V2 seems to tackle 2 massive challenges at once. First, it cracks open those invaluable FFPE archives, making decades of clinical samples accessible for high-res spatial analysis.
Right, unlocking that historical data.
And second, by capturing total RNA with such precision, it delivers this incredibly comprehensive view, including non-coding RNAs and, crucially, this high-fidelity spatial mapping of the immune repertoire, like the BCRs.
Yeah, it genuinely pushes the boundaries of what we can learn from spatial transcriptomics.
Yeah.
Tolerating that low RNA quality, getting the resolution, and capturing the total RNA profile. It makes these vast global biobanks a resource for cutting-edge research in a way they just weren't before. It opens doors for molecular diagnostics, for finding drug targets, really understanding disease in its native context.
So thinking about that last point, the immune repertoire, V2 gives us this unique power to see exactly which B-cell clones, which potential antibodies are physically located right at the site of pathology, be it infection or cancer, within these archive tissues that hold the patient's outcome data. Given that unprecedented linkage, what do you think is the most critical next step for researchers using this tool? How can they leverage the spatial immune mapping to really accelerate the discovery of new targeted immunotherapies?